Okay, everyone, thanks for coming to today's seminar. I'm pleased to, well, if you don't know, Dr. Catheline Froelich, who's a postdoc right now in Titus's lab. Kathleen did her undergraduate work at UMass Amherst and then a master's at the University of Texas in Rio Grande Valley, which is right on the border, before doing her PhD at Australia at,
University of Wollongong.
and then she's been at Dauphin Island, I believe this is your third year.
Almost finished third year.
Almost finished third year. So she's been with us for a while, but she's got a lot of cool stuff to tell us about oyster reefs and cool mutualisms. It should be a great talk. So let's welcome Kathleen.
Disclaimer, I don't study oyster reefs. He did just say oyster reefs, but I don't study them. But I think they're super cool. It's all right. He's got oyster reefs on the brain because that's what he does, and it's super awesome. But I do study reefs and mutualisms. So I'm not going to tell you about everything that I've done in my career thus far fully, but I'm going to focus primarily on mutualisms, which Lee has mentioned. But really, the theme of my research is looking at the functional relationships between organisms and their habitats. So I look at everything from your cleaner shrimp, cleaner gobies. I look at Nemo. My sister threw my face on there, which is pretty cute. We look at gobies that live inside of corals. We look at gobies that live on corals that are cleaning fish. I've also looked at a lot of animals that use artificial habitats, like red snapper, how they use artificial habitats in the Gulf of Mexico, and a bunch of other vertebrates and inverts that use artificial habitats. Oh, 1 sec, I gotta click on this. So when we talk about function in ecology, there are several different questions that pop up in literature. These are not going to be an exhaustive list, but there are a couple questions here that I'll mention. Why do organisms use specific why organisms have specific anatomical or phenotypic traits and physiological traits? Why organisms live alone? Why do they live in pairs, in groups? Why do different species interact? Why organisms have specific requirements for habitats? Why organisms and/or interactions between organisms are important for populations, are important for communities, and are important for ecosystems. And specifically that relationship, those roles that organisms play in communities and ecosystems are also a section of ecology called functional ecology, which is super cool. And then why do several organisms actually co-occur in a single ecosystem and often have very similar functions? That's called functional redundancy. So a lot of different themes of function in science. And I specifically focus on the functional relationships between organisms and their habitats and how that might be important for them, for other species, and for other organisms. Sorry, 1 second. So I'll make sure this works. Aha. So there are a couple other questions that we can focus on when we look at functional relationships between organisms and their habitats. Why do organisms use habitat? It's often used for shelter, maybe shelter from environmental changes, shelter from predators, or it can even be used to actually find prey. Habitat's important for reproduction, for food. How do these habitats affect traits? How do they affect interspecific relationships? How are they important in the grand scheme of an animal's ecology? When we think about habitats, what kind of habitats are we talking about? There are living habitats, there are non-living habitats. If organisms are using living habitats, what kind of relationships are these? Are they mutualistic? So both organisms benefit. Are they commensal, where only one organism benefits? Or are they parasitic, where one organism benefits and the other actually is receiving a lot of costs from this relationship? And then what could actually affect these relationships? Environmental disturbances. There could be human disturbances, so we're impacting these relationships. Other species could be affecting these relationships. And why do we even care? Well, a lot of these functional relationships are important for ecosystems, they're important for conservation, and we really need to understand these to better manage our habitats and ecosystems. So now that I've just thrown in a bunch of questions at y'all, I hope you're excited about function and functional ecology. Let's talk about my research specifically. I'm interested in the functional relationships between animals, their habitats, and ecosystems. And specifically, there are two major relationships that I focus on. So I focus on the relationship between animals and artificial habitats, like sunken ships. Piers also produce habitats in the ocean. We have the Navarre Snorkel Reef, if you know what that is. It's basically concrete blocks that are put down. You can actually get to it from the shore. I do a little project there, which I don't have time to talk about today. My work in my master's degree and all in South Texas was basically focusing on the relationships between red snapper, other fish, and artificial habitats like sunken ships. We had oil platforms, we had concrete culverts that we put out. And it was really cool because that work actually resulted in better management of red snapper populations, better design of artificial reefs, and the largest artificial reef in the US, maybe even in the world, which has been created. They started in 2016, so the last couple of years I was there because I was in Texas for about 6 years for my master's, and then I was a lecturer, and then I was managing a bunch of research students as a research associate. So it's really cool. There's like massive, massive reef now in South Texas where it has a bunch of ships, concrete blocks. It has a bunch of old train track, railroad that are put down. We have a bunch of different structures, and they've been able to actually increase not only the diversity of fish and inverts, but they've also actually been able to make refuges for juvenile species, which are often eaten out pretty quickly. So I'm not going to focus on artificial reefs today. Don't have time for that. But now I'll talk about the rest of my talk today. I'm focusing on organisms and live habitats, and specifically on mutualisms, where both organisms benefit. So for one hand, we've got coral-dwelling gobies, small little fish that live inside of corals. It's a very similar relationship to clownfishes and anemones. You all know Finding Nemo. Hopefully you all know Nemo, right? I also study clownfishes, and then I also study the relationship between client species, so fish and gobies that remove parasites off of other fish in the ecosystem, and their habitat as well. So cleaner shrimp will live on anemones, at least the ones that we study. Cleaner gobies usually live on live corals or sponges. And then you even have your bluehead wrasses actually that can be on any kind of coral head if it's dead or alive. But what I'll be talking about today is how the functional relationships between organisms and live habitats are actually important for their resilience to climate change, as well as being important for some phenotypic traits that they have evolved, specifically looking at clownfish and color patterns. I'll be talking about that. And then also looking at how these functional relationships are important for the ecosystem, where I'll be focusing on those cleaner species and how we actually have a bunch of different cleaner species co-occurring on the same reef. And then I'll be talking about that and how it's important for management and conservation. To do that, I use a bunch of different ecological techniques. And I use everything from looking at behavior, looking at competition between organisms, looking at predator-prey interactions, looking at the effects of season, climate, looking at community ecology, visual ecology, genomics, microbiome, sociality, and so on. And so I have a very integrative ecological background when I look at the functional relationships between organisms and their habitats. As I mentioned, I am only going to be focusing today on mutualisms. Some major topics that I focus on are co-survival, looking at the specific habitat species that are being used, so looking at that host specificity. You have some fish that will only use 1 or 2 species of hosts, and then you have some that use a variety of hosts. I look at sociality, so looking at how many animals are living together in habitat. Are they living alone? Are they living in pairs? Are they living in groups? And why is that important for their functional relationship? I look at the effects of climate change. I look at how the mutualism is key for the evolution of color patterns. And I also look at the functional redundancy. So when you've got, for cleaner species specifically, when you've got species that are that are providing the same function to an ecosystem, why are they co-occurring? Are they truly co-occurring? Are they truly providing the same functions or not in an ecosystem? And why might that be important? As I mentioned in the previous slide, I use a bunch of different techniques. I'll be talking today about how I use surveys, behavior work, visual ecology. I use some manipulative experiments, use genomics. I look at the dietary niche of organisms. I look at their microbiomes. And we also are doing a meta-analysis. I can't actually talk about meta-analysis in detail today. However, we have just started a collaboration with an expert in meta-analyses, and we are going to be looking at how mutualisms are actually geographically distributed in the world. So we're going to see if there's actually a latitudinal gradient with mutualisms overall. Really exciting. It just started. We just had a meeting last week about it. But yeah, so a lot of really cool different techniques that we use for my research. So let's first talk about climate change. I'm sure you all know about this, right? Especially in the last couple of decades, there have been increasing number of heat waves, which have caused increasing number of bleaching events, and an increasing number of cyclones have been occurring. Cyclones, hurricanes, same word, different part of the world. But we've had more hurricanes. I mean, was it last year where we had like hurricanes almost all around us, except for where we are? It was last year, I think it was. And it's just increasing more and more. So we're having more and more climatic disturbances. Our marine environments are heavily disturbed, and there is no stable ecosystem anymore in any marine ecosystem, more or less. And as I mentioned, there are more and more bleaching events that have been occurring because of mass heat waves, especially in the last couple of decades. They have started to see some already in the 80s and 70s, but it's really been in the last decade or two that we've been having more and more bleaching events throughout, where basically there's so much bleaching happening and such long heat waves that corals are just not able to survive. And so bleaching oftentimes nowadays is actually occurring into mass coral death. Now, also, as you probably know, corals and cnidarians, they have a symbiotic relationship with Symbiodinium algae and other organisms as well, which Kenny's lab can tell you all about it. But basically, when there's a long heat wave or cyclones, this symbiosis is under a huge amount of stress. The algae tends to produce a toxin, and basically the symbiosis falls apart. The algae is no longer within the cnidarians. The coral or the algae will bleach. And then what does that actually have to do with the fish that are inside of the corals? If the coral is alive, the fish can stay within. And for some fish, they can actually stay when the coral is dead as well, or the anemone is gone. But most of the time, if there's a long heat wave or extensive cyclones, that symbiosis with the cnidarians and the algae is heavily stressed, and then that symbiosis with the fish and the cnidarian is heavily stressed as well. And so a big portion of my work is to actually look to see if the cnidarian fish mutualisms are resilient or not to cyclones and heat waves. So why do we care about cnidarian fish mutualisms? So cnidarians they provide shelter and breeding sites and food for fish. For corals, it's for coral-dwelling gobies, for example. For anemones, you've got clownfishes. So the fish are receiving shelter, breeding sites, and food. The fish, in return, can actually deter predators. I saw a video, and you probably saw a video, of clownfish attacking the eyes of sea turtles. That was pretty cool. Or the gobies can actually produce a toxin. So they'll shake a lot, they'll release the toxin, and that will actually deter predators, which is super cool. The fish can also remove harmful algae that are around, or they can even remove sand that falls in the coral branches or on the anemones. And then having fish move around the cnidarians can actually reduce the amount of the temperature itself within and around the cnidarians, and that can actually reduce bleaching susceptibility as long as it's not like extensive heat waves, which is super cool. As I mentioned, we know that fish can actually survive. They can survive in cnidarians as long as the cnidarian is alive. There are some species of fish that can actually live in dead corals. I'm not going to be talking about those. But for the species that I focus on, if the cnidarian is gone, the fish is gone. There was a group in Japan, they saw a clownfish. There was just a mass bleaching event. And there was a clownfish like the next day just swimming around aimlessly throughout the reef, about to get eaten, basically. Could not find an anemone, could not find shelter. And when there's no more corals, the gobies are not there anymore either. Now, I mentioned previously the word sociality. What I mean by sociality is a tendency for animals to live in groups. And so it's along a scale. You have on one side You have animals that live alone, they're solitary. And then on the right side, you have those that live in groups. And the extension, like the super extreme of that, is actually when the offspring stay within the group. So it's this big family unit. And so when they live in groups, they can actually help to take care of their habitat, which is really key. We see that for corals that have more gobies in them, they actually have more care of the coral, and so the coral can actually grow bigger. Same for anemones. Anemones are bigger when there are bigger groups of clownfish because clownfish are able to maintain that anemone better, make it healthier. Now, for coral gobies and for clownfishes, they live within a social hierarchy. The two biggest fish breed. For clownfish, the female is the biggest. For gobies, depends on the species, and they can actually interchange. So gobies can actually go back and forth between female and male, even though it's very taxing. Clownfish, they can go from male to female, which is super cool. But the two biggest are the dominant, they are the breeders, and the others are usually non-breeders. They're subordinates. And their job there is to help maintain the habitat, maybe even take care of the eggs, but not usually, but they help to maintain the habitat and stay out of the way of the big guys so that the big guys can then breed. And then if one of the two dominants dies, the next rank below is going to move up. But they have to control their size. So if the subordinates, the non-breeders, are getting too big, they can get evicted out because they're then threatening that position of the next higher up, which is super cool. And because they live in groups, they've actually modeled for clownfish, there's a group that went every single day, visited groups for a whole year every single day, and they modeled mortality, growth, and so on. And so clownfish can live up to 30 years, which is super cool. Which, if you think about like bees and ants, the queen can actually live like several years, even up to a decade or two, depending on the species, because they're living in that family unit. So sociality is really important for my species, they're important for the fish, but they're also important for those cnidarians, because bigger groups, more maintenance, healthier cnidarian, bigger cnidarian as well. All right, now let's focus real quickly on the coral fish mutualism that I focus on. So I focus on coral-dwelling gobies. They are from the Gobiodon species. They live inside of Acropora coral species. There are over 20 species of Gobiodon. They are super cute. They vary in color, shape slightly. Some have polka dots, some have small polka dots. I know people say that gobies aren't cute, but I mean, like, these are super adorable. Super adorable. And they are super small. They are between 1.5 centimeters to 6 centimeters. So one might get to the length of your finger, but like most of them are as wide as your finger. They're super small, or as wide as your thumb, maybe. That's more like 1.5 centimeters. They're super small, high predation risks. So they need that coral to survive. And also, they have preferred hosts. So several species have a preference for one host. And then other species use actually a variety of hosts. But if they have preferred hosts, they will then have evolved to have morphology that's better to be able to maneuver within the coral. They can actually get bigger. So oftentimes you might see a specialist goby where it's actually like a lot higher. If we look at fish height, they're a lot higher and skinnier, so they can like maneuver within the branches. And they're bigger because of that, so they can actually outcompete other species. So there's a benefit to having a preferred host, or there's a benefit to using a variety of hosts. Let's say there's a lot of disturbances, you don't always have your preferred host around. It's a huge benefit to actually using a variety of hosts. Now, let's talk about how cnidarian and fish mutualisms respond to climatic disturbances. So that is what's been a big part of my PhD and ongoing work as well. Mutualisms are really important for the fish and for the coral, and we know that coral are building blocks of coral reefs. And the Acropora species of corals that the gobies are found in are often fast growing, but they're also some of the first ones to actually bleach and die from climatic disturbances. But they can also, some studies have shown that they can actually grow back pretty quickly, the corals. So what about the whole mutualism? What about the fish-coral mutualism? How is that relationship affected by climate change? And what could be the potential mechanisms from the fish perspective that might explain some of these responses? So I first set the scene. I did a population monitoring before and after several cyclones and bleaching events. It's not me, it's a massive team. I'm not going to act like it's just my project. And then we looked at potential mechanisms from the Gobi's perspective. We looked at that host plasticity. Is it able to change its preferred host? If it preferred one host always, can it actually start using a bunch of hosts or the reverse? What about social plasticity? Looking at group size, can they actually alter their group size? Looking at movement plasticity, if we move fish around, so let's say, for example, with a cyclone, right? There might be a lot of displacement of fish during cyclones, and so a fish might end up somewhere else. What's it going to do? Is it going to use a new coral? Is it going to go back to its preferred coral? And then we looked at population structure. So before and after disturbances, how did populations genetically, were they still quite diverse? Were there genetic bottlenecks? What happened? And all this is important. I got a little cut off. Sorry. This is also important when we think about the plasticity. So how are the mutualisms able to survive with climate change? Maybe they inherently have a lot of abilities to actually be flexible, to be able to handle continued effects of cyclones and bleaching, maybe not. So when we think about the word plasticity, we're talking about the ability to change particular characteristics or behaviors in order to accommodate for different conditions. So from my perspective, I'm talking about climatic disturbances for cyclones and bleaching. So we did a lot of observational surveys before, after cyclones, and after bleaching events at one location. Corals bounce back pretty quickly. Gobies do not. They are not resilient at all. And so let's talk about this briefly. How do we do this? Lizard Island is in the Great Barrier Reef. And Lizard Island, I'm actually going again in December. I'm super excited. We're going to keep monitoring these guys. Lizard Island is this amazing field site that's been decades and decades and decades established since the Australian Museum. It is probably my favorite. It's the most technologically advanced research station I've ever been to, and I've been to a couple. And people have been going there for studying a bunch of different things. And unfortunately for the reef, fortunately for scientists, kind of fortunately, there have been massive, massive climatic disturbances. So in 2014, Cyclone Eta was Category 4. It directly went over the reef. So we surveyed gobies just before. Then there was a massive cyclone. It literally went like directly over the reef. Then the next year, we surveyed them again. And then there was a cyclone that went the opposite direction, directly over the reef. I think it was Category 5 this time, Cyclone Nathan. Again, cyclone means hurricane, right? In Australian lingo. Then we went back the year after that. And then there were two mass bleaching events, the biggest bleaching events that have ever hit Lizard Island. They made news globally. You've probably heard about them. It was an absolute disaster. The reef did not look the same at all. And then we returned. This is the first time I joined on the trip in 2018. It was honestly devastating and just quite horrible to see. There were corals here and there. They were tiny. They were newly settled if we saw corals. Once in a while, we saw really big corals, but basically there were very few corals. There was coral debris everywhere. It was really depressing, and I'll show it, but basically there were very, very, very few gobies around. And if there were corals, the corals were really tiny. Just before then, just around that same survey year is when we started to monitor clownfish populations. So clownfish, anemone mutualisms there. 2018, 2019 was safe from bleaching. I realize I don't have the drawings here, but they were basically like starting 2020, there was mild bleaching practically every year, ever since then. We went back in 2020, surveyed the clownfish and the gobies. I went back in 2021 to look at the gobies. 2022, surveyed the clownfish and the gobies. And then we just booked our flights. We are going Adelaide. One of the boat's interns is joining me in November, December to monitor gobies and clownfish again. So it's just really cool. We've got this really cool long-term monitoring programs now that we've been establishing for the coral goby mutualism and for the clownfish anemone mutualism. We've used Transx for gobies, and we were trying to actually like individually monitor the same groups of gobies over the years, but after the cyclones and after the bleaching events, all the groups that we had monitored disappeared, AKA the coral died and the gobies were gone. So we've just continued maintaining the coral goby dataset with Transx. And then with clownfish and anemone mutualisms, they're a lot more resilient so far. So we have been monitoring the same groups over and over again. However, the Red Sea was supposed to be this like safe haven from bleaching, if y'all know. Recently in the Red Sea, the last 2 years, there have been mass bleaching events, and now there's like only 20% of the clownfish anemones left there were previously, which is devastating. I promise this is not going to be a totally depressive talk. I promise. But yeah, so I'm curious to see this year now coming back where there have been multiple bleaching events recently, how the gobies and the clownfish and anemone mutualisms are faring. All right, so some quick results. This is for gobies. We've got the disturbances, cyclones, and bleaching events here. All right, so when we look at coral richness, the number of coral species, it dropped a little bit after the cyclones, dropped immediately after the bleaching events, but then a couple years after those mass bleaching events, it's actually higher than it was before. Cool, interesting. And you can see also I've got a couple symbols there so you can follow along. What about gobies? The number of species was pretty consistent over the cyclones, dropped after the second cyclone, dropped even further after the bleaching event and hasn't recovered. Looking at coral size, corals became extremely small after bleaching and are still extremely small. What about goby group size? They were living in groups before cyclones. They started living in pairs only after cyclones. After bleaching events, they were by themselves, basically. And a couple years after, they were still more or less by themselves. And that's because the corals were gone, the gobies were gone after the bleaching events. And so these are juveniles that have settled. But even so, 3 years after, they should be in groups, they should be reproducing already, they're still not recovering. Then we look at how many of the corals are actually occupied. Almost all of them were occupied before disturbances, decreased a bit after cyclones. After bleaching, 1 out of 4 corals are empty, and they continue to be. And so corals are doing better than the gobies, but the corals are extremely small. So yes, there are corals, but they're extremely small, which is not great either. And so they're also still recovering quite slowly, even though a cropper is supposed to be fast growing. And as I mentioned, looking at the clownfish anemone mutualism, we are adding more data in this year, so I don't have any data yet for that. All right, so we've set the scene. Corals are doing better than gobies, but even then, not great. Then we looked at host plasticity. We see there is some host plasticity. The few gobies that actually remain sometimes are able to actually switch to the newly abundant host. Is that a good thing or not? If they're not in their preferred host, they might not be able to grow as fast. They might not be able to maintain the coral as much, but at least they're able to change the host. So I'm going to very quickly, this is a bit of chaos. It's a really cool figure. It looks a bit chaotic, but let me explain it to you briefly. On the left-hand side, on the vertical axis, you've got all the different goby species. All right, okay. 2014, vertically 2014, you've got the different corals that are being used. So the very top is Acropora acerealis, barely used. Acropora nisuta, a bunch of goby species are using them. Acropora tenuis, a couple of goby species are using them. And there's also some using Gemifera, Loripes, Millipora, Ballida, and so on. Okay, 2015, 2016, we're seeing similar patterns. Then, so those are after the cyclones, then those mass bleaching events happened. We see that most of the gobies are actually switching to Acropora cerealis that was barely used previously, which Acropora cerealis now has become the most abundant coral, and gobies, a lot of the species are able to actually switch to Acropora cerealis. And that remains so in 2020 as well. So they're able to switch their hosts, at least for the gobies that are remaining. I haven't really shown it here, but Gobiodon bilineatus disappeared. Gobiodon regiolatus, which is not on here. Oh, actually, it is on here in gray. I saw like 1 or 2 of them after the bleaching events, even though they're like super abundant. So we're not seeing, I think it was overall out of the 15 species that I surveyed, 6 of them disappeared fully after the bleaching events. They were still there in the cyclones, but they disappeared and they haven't come back, or I've just seen like 1 or 2, super rare. All right, so then we looked at social plasticity. What's happening to that group size? So there are smaller groups, and as I mentioned before, they're living alone, primarily after bleaching, and there's a couple that are starting to live in pairs. That's not necessarily the best thing. There are less fish to help maintain their coral, but also if they're living alone, they can't actually reproduce. They don't move around a lot. They really don't move around. They stay within their coral. So they're not able to reproduce, which means that there's a lot less of a turnover and abilities for producing a bunch of larvae. So again, like I mentioned before, it's really important for not just the fish, but for the cnidarian to have fish living inside of them. So this is they did a study for clownfish and for gobies. They looked at the relationship. The biggest individual, that size of that biggest individual, is directly related to the anemone size and the group size for clownfish. So biggest clownfish, the bigger it is, the bigger the anemone is, and the most number of fish that can live inside of that. The same is true for coral. So it's really important for both fish and for the cnidarian to have groups. So basically, we first looked at species. There are some species that tended to live in groups, in pairs more often, even before disturbances, but they're still in pairs able to reproduce. We looked at whether they were staying in pairs or groups. So we looked at a sociality index, basically 0.5 value. Above that, they live in groups. Below that, they live in pairs. So if they lived in pairs before disturbances in solid, they still live in pairs after disturbances in dashed bars. I'm going over this very fast, but you get the general idea, staying as pairs. What about groups? If they're living in groups before disturbances, one or two were able to live in groups, but then a lot of them actually weren't able to live in groups after disturbances. So pair-forming species often stay pair-forming if they actually can find a pair. And then group-forming species after extreme disturbances, they usually are not able to live in groups, and only a couple of them can. Then we looked at movement plasticity. Basically what we did was we wanted to recreate kind of this idea of cyclones. When fish are during cyclones, they might be moved around. What do? Do they go back? They try and find their original host or not? So we took a fish, we tagged it. We took a fish, we tagged it, we moved it to a coral skeleton right next to a live coral. So it had the chance to sit in a dead coral or to move to another coral right next to it. It did not try to stay in the dead coral, did not try to find the live coral right next to it. It actually went home. So either they died or they went home. They basically disappeared entirely and they tried to find their way home. So that they are still willing to face high costs of movement to find their preferred host. So that initial host that they settle in is really important for them. Then we looked at population structure. I don't have time to talk about this in detail, but basically we first looked at 3 different areas. So we looked at Lizard Island, which I've been talking about. We looked in the north in Papua New Guinea and in the south in One Tree Island. We found that there's a lot of population structure. So there's not much mixing going on between populations, even though Papua New Guinea, Lizard Island, and One Tree Island are respectively pretty close when you look at the giant ranges of Gobiodon. And then we looked before and after the cyclones and bleaching events in Lizard Island. So the species that we studied, 2 of them experienced extreme genetic bottlenecks. So the diversity after the disturbances was extremely low. One of the species continually had very low genetic diversity, and it's actually a threatened species. And the 4 species managed to maintain its genetic diversity. This is currently in review. I just submitted it a couple weeks ago. So in conclusion, the mutualism between coral and gobies is not functionally able to stay the same and be resilient to climate change. And this at least is for gobies, right? I'm going to be looking at clownfish as we continue to monitor them. So some future directions of this work: how do clownfishes respond? And also, because the coral goby mutualism is really not resilient at all, when we think about how coral reefs are responding to climate change and to bleaching events, scientists have shown that it's going to take a really long time. It's going to take decades. And so I believe that actually the coral goby mutualism could be an excellent indicator of reef health. So my next step is to then be able to look at this mutualism and then be able to use that In the future, people could just monitor coral and gobies, and that could give us a good indicator of reef health. So that's my next step in this study. Okay, now switching gears to something less depressive. I also, for my postdoc, look at how the mutualisms between anemones and fish are functionally responsible for the color patterns of clownfish. There are 3 distinct color patterns that have converged with host use. So the question is, has the ecology converged? Basically, we know there are 3 color patterns. 2 of the color patterns are for clownfish that are host specialists, so they specialize on one anemone species. And then a 3rd color pattern is used for generalists. So they use a variety of different hosts. So why have they evolved these color patterns with their host use? Why is that mutualism key? We need to look at the whole ecology of the fish and the anemone to better understand that. So we looked at their behavior. How far are fish willing to move from the host? What does a predator see? What is the fish eating, and how is that related to their anemone? Are they eating a more benthic diet? Are they eating a more pelagic diet? What does their microbiome look like? Because fish, and they've shown that clownfish and anemones, their microbiome can actually converge. So they can actually have very similar microbiome because they're living so close to each other. And then what about when you have some fish that are supposed to be specialists, but they once in a while will use a different anemone species? So why is an animal coloration important? So what's its function? It's important for crypsis, so for camouflage. It's important for blending in. It can be used, actually, there's a form of it called disruptive coloration, which can be used for zebras. So zebras are super striped. When flies try to land on them, the flies are moving around really a lot, and so the stripes on the zebra are actually really confusing the fly, so the fly can't actually land on it. Animals can use coloration to communicate socially. They can be used to signal a toxin, a poison, a venom, also known as aposematism. They can be used for sexual selection. And they can also be used, it's pretty cool, they can actually be used to reduce abrasion, to make them animals more thermal, keep them warmer, for example, in winter times. They can also be used to be more aerodynamic. So for clownfish, as I mentioned, there are 3 major color patterns. You've got red clownfish that have specialized on Entacmea quadricolor, like Amphiprion rubicinctus. You've got orange clownfish that specialize on Radianthus magnifica, like your Amphiprion paradorion here. And then you've got your host generalists. They are black with really thick white stripes, think zebra. And they use a variety of hosts. So I wanted to know, why do these colorations converge with host use? We looked at their behavior. Do specialists stay closer to their hosts than generalists? Looking at visual acuity, how does a predator actually see these color patterns against their anemone background? Are specialists less visible? Are they able to camouflage better? Looking at the diet, do specialized specialist species, so those orange and red clownfish, do they eat a more specialized diet? Do they eat more benthic diet, more what's around the anemone, versus the generalists? And what about their microbiome? Is their microbiome also more specialized for those orange and red specialist species? So we looked at their behavior. We basically filmed these. It was quite fun. So I'm going to just play these videos here very quickly. This is a GoPro. We use GoPros, so this is a 2D camera. And what we did was we basically measured the body depth of fish in every minute in a frame, and we measured how many body depths away a fish is from its anemone. So you can see the generalist in the video to the left, and the bar in the graph to the left, you can see it's super colorful. So the fish was almost never in the anemone in gray, was within 1 body depth in blue, 5, 10, 20, and over 20 body depths away, which is like super far away. So at that point, they don't really care about predators. They are super far away from their shelter, from their anemone. What about specialist species? They are either in gray in the anemone, 1 body depth away in blue, or once in a while a little bit further away, but they basically stay within or around their anemone. Then we wanted to see, how does a predator see this? So we're first going to play a quick game. We're going to have you guys count how many clownfish you can actually see first. So we're going to test it from our vision here. All right, so I've got a picture here. Put your hand up and show me with your fingers how many fish you see. A lot of you are right. There are 3 in here. This is a red specialist. I'm impressed for those that saw 3. Maybe they're lying. I don't know. Somebody saw 4. Great. Amazing. I love it. There's a lot of algae in here. This is actually a more or less dead looking reef, as you can tell. But there's anemone in there. What about here? How many clownfish do you see? I tricked you. Most of these are actually domino damsels. They are not clownfish, but there is a clownfish in here. There's one. And it blends in super well. It's an orange specialist. What about here? I threw an domino damsel in there, a tiny one, just to throw you off. But everybody's right. There's two in here. This is the generalist. It sticks out a lot, easier to see already. So we can see it. What about a predator? Oh, that looks funky. We took pictures of the fish with a color checker on it so we could actually measure the accurate colors of the fish and the anemone. Threw it into a fancy program, all free in ImageJ. Ask me if you want to know about it. And we basically looked at, wow, this got really fucked up. Sorry. I changed the font size and it messed a lot of things up. Wowza. Anyways, okay, so I'm going to show with the pointer. Okay, so on the left-hand side is luminance. How bright is the fish compared to its anemone? The generalist sits here. The specialist's mean for the orange specialist is down here, and red specialist is down here. So the generalist is a lot brighter than the anemone. And then you can see on the right-hand side, we looked at the actual color itself. So the actual color, not just how bright the fish is. And the generalist color is a lot more different to its anemone than the specialists. So basically that means that the specialists are less bright and less colorful than their anemones. So they blend in better. Okay, then we looked at the dietary niche doing stable isotopes. I don't have time to go into a lot of detail, but basically x-axis values to the left mean they're eating a more pelagic diet, values to the right mean eating more benthic diet, and up and down is the trophic level. Higher up values, they're eating in a higher trophic level. So that's like your herbivores, carnivores, higher predators, and so on. In orange and red, the specialists are eating a much more benthic diet, and their ellipses are a lot smaller. So they're eating a much more specialized diet, where the generalist has much wider ellipses and is eating much more variation between benthic and pelagic diet. Then we looked at the microbiome, and without going into much detail, okay, so in blue and green are the anemone species. The microbiome of the anemone species are basically fully enclosed within all the clownfish species, which is cool. Okay, so there's some convergence there. Shocker. There's a mutualism. Awesome. But what's more interesting is that the generalist in black, that ellipses is a lot bigger. So there's a lot more variation in the microbiome of generalists than there is in the two specialist species. So if we put all of that together, all right, so we know that there are two host specialists, the red specialist and the orange specialist, and then you have those black fish with really thick white stripes, think zebra. They use a variety of hosts. Behaviorally, the two specialists stay near the anemone. The generalist doesn't care about their anemone, almost. Sometimes it like runs back to the anemone once in a while, but basically is never in the anemone. Visually, the predator has a much harder time to see the fish in the anemone for the specialists versus the generalist. Diet-wise, the diet of the specialist, it's much more narrow and more benthic than that of the generalist. And the microbiome is much more specialized for the specialist than the generalist. Okay, so what does that mean? That means that not only are the color patterns converging with the host use, but the whole ecology of the fish is also converging with host use and with color patterns. So that's super cool. So really seeing that evolutionary trend with host use, with diet, with all the ecological tools that I've looked at. So now we can predict the function. For specialists, we predict that they're actually camouflaging within the anemone. And ecologically, it's all lining up. It's all making sense. And then for the generalist, either they are aposematic. So they're either signaling that they are toxic, or more likely their anemone is toxic. So anemones are super toxic, which I've gotten stung. It makes little red bubbles. And from Finding Nemo, right, the anemone stings. But the clownfish has this like protection against the sting. So either the fish color pattern is signaling I'm toxic myself because it's super dark, which oftentimes that can signal a toxin, or it's that zebra effect, disruptive coloration, where if a predator goes to strike a clownfish, that motion of the fish with the stripes is confusing the predator. So the predator doesn't actually know where the fish is. So we need to do some future work to really differentiate those two. And we're also comparing a bunch of different species in the genus just to confirm everything we're finding here. But it's super cool. And then Abby Nguyen was an RE student this summer, and I'm not going to go into detail, but she basically looked at host plasticity. So when specialist species were actually found on a different host species than what we think they're supposed to, what happened ecologically? So there was some phenotypic plasticity for the orange specialist. It was actually a little bit darker in a different host. And then for the red specialist, it actually was further away from the host. So some kind of ecological plasticity that we are seeing. We haven't fully figured out why that is and what these functions of these different aspects of their ecology are, but we do see that there is some plasticity with host use if they don't find their preferred host, which hopefully is good, right? With these environmental disturbances, hopefully they can actually find different hosts if that's just how it happens. Moving on, we're going to switch up to now cleaner species. This has been much more recent focus of my work. So we are looking at cleaner shrimp, cleaner fish that remove parasites off of other fish. And these cleaner species are usually associated with habitat. So cleaner gobies are found on corals, sometimes sponges. And cleaner shrimp are found, especially these cleaner shrimp at least, are found on a species of anemone. And so there's this relationship between the habitat, the cleaner species, and the client species. And functionally, the cleaner species can use their habitat to actually advertise. Because yes, I've blown up the pictures of the cleaner shrimp on the bottom right, but it's translucent. It's extremely hard to see when it's in the anemone. It really blends in. But the shrimp is using its color pattern and it dances. And so dancing against its habitat, it's able to signal to clients, hey, I'm ready to clean. Come over here. You can see me. So it gets cleaned. And then for gobies, you can see they're a little bit easier to differentiate from the corals, but they use a variety of different coral hosts. So they're host generalists, and sometimes they blend in a lot. So sometimes they usually stick around and they just wait for a client, but once in a while they might have to use different strategies. But basically there's this functional relationship between all these three players. And then you also have these bluehead wrasses in the top right. We find them dead coral heads, live coral heads, but they use habitat, but much more loosely. But they can actually still clean clients. So this is important. These functional relationships are really important for ecosystem. This is a great slide that I took from NOAA website. But they're basically nicknamed coral housekeepers. They're really important for cleaning. They're especially important for cleaning herbivores, which often in algae can get a bunch of gunk on them or parasites on them. Maybe they're eating in sand. Parasites like to live in sand. And so the cleaner species are able to remove those parasites. They're important for fertilization. They're important for pest control. They can even protect corals from other fish that are trying to eat the coral. And then in return, the cleaners are getting shelter from the habitat, but they can also help to just overall, because they're doing all these ecosystem services, they can improve the reef habitat, they can remove parasites, they're ingesting parasites, so they're removing that. And they can also just overall affect and improve the diversity of reef communities because they're able to remove parasites and remove other things that are bothering client fish. So briefly, I'll talk about Corey Brown. She's a master's student. Some of you know she's just graduated recently, which is very exciting. She studied cleaner shrimp. And so very quickly, she basically studied the same cleaner shrimp stations, we call it a station, a cleaning station, on a weekly basis. So for 5 weeks, there were less stations that we filmed the first week. That's why there's a lot less in week 1. But basically, the amount of time that a cleaner cleaned a fish was roughly consistent every week. And the cleaning rate, so how many times did they actually clean in an hour, was roughly consistent on a weekly basis over the same reef zone. But the same station wasn't always popular consistently. So on a reef level, cleaning is consistent, but when you look at a station by station level, there's actually a lot of variation. So maybe clients they just have like a bunch of different stations in their back pocket that they like to visit throughout the week and throughout the month. And then we just took a class to Honduras, same site as Corey's project, in May. It was May. No, in June. No, it was May. the May semester, the coral reef ecology class for DISL. We went there in Honduras. We had 7 undergrads and 2 master's students. They learned all about coral reef ecology. It was really cool, but we also actually have 2 papers that we can publish from it. And so it's really exciting for undergrads to get that experience. And we focused on functional redundancy, which I briefly mentioned previously. We have 3 cleaner species that are co-occurring on the same reef: cleaner gobies, cleaner shrimp, and those bluehead wrasses. Why are they co-occurring? And are they truly all having the same functional roles in every single part of the reef? So are they truly functionally redundant or not? Why would that be important? Well, when environmental disturbances hit, right? One cleaner species might be hit a lot harder than others. So if one cleaner species disappears or becomes extremely rare, there's the opportunity for the other cleaner species to take over their stations or to take over the general roles. So this idea of offsetting that disturbance. So we see here in blue, the cleaner wrasses on the left-hand side. They're cleaning, especially in the spur and groove area. It's the shallowest part of the reef. They're cleaning the most. They're doing a lot more cleans, but they just jut in there, basically just pick and run back off. So cleaning them, they clean for a lot shorter time for each interaction. For cleaner shrimp in gray, they clean a lot less frequently, but for much longer. And then gobies are roughly in the middle there. And we're seeing also differences in different parts of the reef. So they might be functionally redundant to a certain extent, but there is a lot of variation still there. Future work, we are actually combining with a bunch of different research groups. And we have now got decades-long data for many different cleaner species in the Caribbean and the Red Sea. And so we are going to be comparing cleaner species in the Caribbean versus Red Sea, as well as in the Caribbean for those long-term monitoring. So how has the cleaner relationships been affected throughout the years? And so with that, I'll leave you my summary slide of everything, and there's a couple minutes for questions.